Welcome back to this next video in which we are studying headaches. Okay, so we're currently in the process of discussing cluster headache, which remember is the name for the disorder where you are susceptible to suffering from cluster headaches. And we now want to discuss the pathophysiology, or what is currently thought about the pathophysiology that underlies cluster headache. So before we begin this story, again allow me to give you the warning that I've given you previously in this video, which is that again what we're about to discuss is controversial. I can only give you our best current ideas about what underlies cluster headache, the pathophysiology behind it, and there may well, and indeed there is, holes in the story that I'm going to tell you, so be aware of that before we actually begin this discussion. Okay, so let's now begin the story of the pathophysiology, or what is believed to be the pathophysiology underlying cluster headache. So it's helpful to remind ourselves of what happened in migraine. So remember, migraine was the name for the disorder where you were susceptible to suffering from migraine headaches, and it was fundamentally a brain disorder. It was a disorder of the brain that meant that the brain was susceptible to a certain brain phenomenon occurring episodically, and that brain phenomenon that occurred in migraine was cortical spreading depression. And when you suffered from an episode of cortical spreading depression, that would lead to suffering from a migraine headache. So migraine was the disorder that made you susceptible to a brain phenomenon, cortical spreading depression, which when that occurs, gives rise to the migraine headaches. Cluster headache is very similar. Cluster headache is the name for the disorder, and it makes the brain susceptible to a certain brain phenomenon. So again, it's believed to be a brain disorder, and the specific portion of the brain which is susceptible to a brain disorder, a brain phenomenon in cluster headache is believed to be the hypothalamus. The hypothalamus is believed to be incredibly important in cluster headache pathophysiology. One, because of this circadian and circannual rhythm business here, and also because we can actually see that something weird is happening in the hypothalamus when someone is suffering from a cluster headache. But I'll say more about that in just a moment. So, in cluster headache then, you have a hypothalamus that is susceptible to a certain brain phenomenon. Now, what is the brain phenomenon that can occur then episodically in the hypothalamus in someone with cluster headache? Well, it appears to be hyperactivity, far too much activity, potentially epileptic or epileptic-like hyperactivity. And this hyperactivity, it occurs when the cluster headache actually occurs. We can see it occurring if you do brain scanning, functional brain scanning on someone who is currently suffering from a cluster headache. We can see this overactivity in one side of the hypothalamus, and the side that is affected corresponds to the side where you will feel the headache on. Okay, so let's discuss this more in detail now below. So to discuss this further, let me draw a picture of the hypothalamus. So the picture I'm going to draw is a picture of the brainstem and the diencephalic structures. And remember, the diencephalic structures are the thalami and the hypothalamus. And we will be viewing this from the left-hand side. So let me do this here now. So we'll start off with the pons, and then the midbrain below, and then of course you'll have the spinal cord below that. So those are the lower brainstem structures. Then above the pons, of course, you're going to have the midbrain, like so. And then, of course, on top of the midbrain, you've got the two thalami on either side, the left thalamus and the right thalamus. We are drawing a picture here viewed from the left-hand side, therefore we will just see the left thalamus, which I'll mark on here now. And remember, the thalami are both egg-shaped structures, like so. So there is our left thalamus, and now I want to put on the hypothalamus here. Of course, we're seeing the left side of the hypothalamus, and I'll do this in green, like so. So here is the hypothalamus in front, and you have structures dangling down from the hypothalamus, of course. You have the optic chiasm there at the front, and then the pituitary stalk with the pituitary gland there, and then the mammillary bodies. So just going over those again. Here is the optic chiasm that I'm now colouring in green there. cut through, of course. Then we have the pituitary stalk here with the anterior and the posterior pituitary there, and the left mammillary body here. Of course, there's also a right mammillary body on the right-hand side, but we're looking from the left-hand side, so we'll see the left mammillary body. So here's our picture of the hypothalamus. Now remember, the hypothalamus is hollow. The third ventricle runs through the middle of the hypothalamus, splitting it into a left side, which is the side we're seeing, and a right side, which makes up the two lateral walls of the third ventricle, which then goes through the middle of the thalami too. Okay, right, so here's the picture of the hypothalamus. In cluster headache, if we have someone with cluster headache, something will be different about their hypothalamus that makes them susceptible to hyperactivity episodes occurring in one side of the hypothalamus. And between different episodes, the different episodes can be on different sides of the hypothalamus. So one episode might be on the left side of the hypothalamus, the next episode might be on the right side of the hypothalamus, and those will give rise to cluster headaches moving between the different sides. So when you've got hyperactivity occurring in the left hypothalamus, it will lead to a left-sided cluster headache. And if you have the hyperactivity occurring in the right hypothalamus, that will lead to a right-sided cluster headache. So let me just mark this on here. So I'm going to show this in a really crude way. So just as this sort of pink explosion here, that's representing this hyperactivity, and this may well be epilepsy-like. So this is hyperactivity occurring in one side of the hypothalamus. And as I say, you can see this occurring if you take someone who suffers with cluster headache and they're currently suffering from a cluster headache at that moment, and you put them in brain scanners, you can see that the one side of the hypothalamus, the side that is on the same side as where the pain is completely overactive. So this is one of the major pieces of evidence that the hypothalamus might be crucial in the pathophysiology underlying cluster headaches. Okay, right. So now turning back to circadian rhythm and circannual rhythm. So these obviously also fit with the idea that maybe the hypothalamus is extremely important, because the hypothalamus is an absolutely ancient portion of the brain, evolutionarily ancient. Loads of other animals have a hypothalamus, and it does very similar roles to what it does in humans. So, mice have a hypothalamus that does pretty much the same as the hypothalamus in humans, even though they don't have the incredible, massive cerebral hemispheres that humans have, which do all the incredible things that make the human mind unique. The hypothalamus is not an absolutely incredible part of the brain. It's not one of these really, really advanced bits that makes us human. It's one of the evolutionarily ancient parts of the brain, which is responsible for very, very basic functions of the brain, very, very crucial functions of the brain. And it is believed to be where there is a circadian clock, a clock that is capable of making sure that the brain knows what the current time of day is, and it probably also contains a circannual clock. Now, we know where the circadian clock is. We have a very good understanding of where the circadian clock is present in the hypothalamus. We don't really know at present where the circannual clock is, but it's very likely that the hypothalamus also contains a network of neurons that are involved in working out what time of the year it currently is. And these sort of things are useful for animals to know, so that they know when they should be asleep and when they should be awake. That's obviously the circadian clock's function. And also the circannual clock, some animals hibernate, so they need to know when to hibernate, so it's useful for a part of their brain to be clued on to what time of the year it currently is. So let me just give you the background of where is the circadian clock. So the circadian clock is in two nuclei called the suprachiasmatic nuclei. So big, big names. So these are hypothalamic nuclei, suprachiasmatic nuclei. And for short, we can abbreviate the suprachiasmatic nuclei down to the SCNs. So where are they? Well, you might be able to guess where the suprachiasmatic nuclei are. Suprachiasmatic. Supra means above. Chiasmatic clearly refers to the optic chiasm. So these nuclei are going to be just above the optic chiasm, and indeed they're in this sort of position here, and you have two. This, of course, is the left one that we are looking at here, but there would be one sat next to it, also above the optic chiasm, which would be the right suprachiasmatic nuclei. And really, it wouldn't be visible. I'm just showing its basic position, but they're above the optic chiasm. It's probably helpful just to draw a picture looking from the front, a basic picture looking from the front. So if we were to look from the front, here is the anterior wall of the hypothalamus, making up the anterior wall of the third ventricle behind. Here is the optic chiasm, which I'll colour in here in green. And then above the optic chiasm, we have these two nuclei. There is the right one, and here is the left one. And these are the suprachiasmatic nuclei. And these are involved in keeping time within the brain. So they are what we refer to as the circadian clock nuclei. Now, how do they actually work? Well, they consist of loads and loads of neurons, of course. They're nuclei fundamentally, so they're collections of cell bodies inside the central nervous system. So there are loads and loads of neurons in each of these nuclei, and those neurons, their firing rate depends on what the time of day is. So during the day, during the light period of the day, they will be firing at high rates, whereas at night they are firing at low rates. So their rate of firing is gradually changing throughout the day, and if we were to draw a little graph of this, it would be sinusoidal. So what we can do is indeed draw a little graph like so. So let's have this as time, and let's put some hours. So let's have this as 0 a.m. in the morning, and we'll do, what's a nice one to do? We'll do every 6 hours, let's say, which will split nicely up into 24. So then we've got 6 a.m. in the morning, then we've got midday, 12 o'clock, then we'll have 18, which is 6 p.m, and then of course it'll be 24, which is back to midnight. Okay, and on the y-axis here, we're putting firing rate of the neurons in the suprachiasmatic nuclei, and it's going to fluctuate in a sinusoidal way according to time. So let me show this. So at night it's going to be quite low, and then as we go to morning it's going to be gradually going up and up and up, and then it will gradually go down as we approach night again, and there we are at 24. That's imagine that I can put in the 24 there and it's gone back to where it was beginning. Obviously I haven't quite managed to get them at the same levels, but imagine it is. So it goes up during the day and then it comes down. So it does this day in, day out. It follows a rhythm that lasts 24 hours. It's a circadian rhythm, and this controls loads of other things in the body that also need to be tuned to a circadian rhythm. So the output of the suprachiasmatic nuclei controls loads of other things. So for instance, cortisol level in the blood also goes like this. It's higher during the morning and then goes down as we approach the evening, etc. Loads of bodily things fluctuate on a 24-hourly basis, and they're coordinated by the suprachiasmatic nuclei, the circadian clocks in the hypothalamus. Okay, so the suprachiasmatic nuclei, they are believed to be the circadian clocks within the hypothalamus. We believe there is probably a circannual clock present in the hypothalamus, some bit that is involved in working out and telling the rest of the brain what is the current time of the year. However, we do not know the identity of that circannual clock, but it probably is present within the hypothalamus. Now, why have I gone into this? Well, of course, we know that cluster headaches, they follow these circadian rhythms within a cluster, and the clusters follow circannual rhythms. So that means that this hyperactivity in the hypothalamus, if this is what truly underlies cluster headaches, and according to this theory, this is what underlies the cluster headaches, then this must only be generated at the right times of day and at the right times of year. So this is why it kind of all fits together. The hypothalamus is this site where utterly we know what the time of the day is, and we most likely know what the time of the year is. And then somehow, at certain times of the day and certain times of the year, which is information that the hypothalamus knows, somehow in people with cluster headaches, that then leads to the hyperactivity. Somehow the hyperactivity in one side of the hypothalamus is then generated. But I can't give you a more full story than that. That's, I'm afraid, the holes that I'm going to have to leave in this story. We believe this hyperactivity in one side of the hypothalamus is going to be the episodic brain phenomenon that underlies the cluster headaches. When is it generated? We don't know, but somehow the circadian and circannual inputs, which are information which we believe the hypothalamus has, must somehow feed into this. And then only when the time is right, when the time of the day is right, and when the time of the year is right, is this brain phenomenon actually generated. So, at certain times of the year and at certain times of the day, in those times of the year, this brain phenomenon, this hyperactivity in one side of the hypothalamus will be generated in people who are susceptible, i.e. people with cluster headache, and then it's going to actually lead to the cluster headache. And what we now want to discuss is how is it going to lead to that cluster headache. Now, it's going to lead to the cluster headache on the same side as the side of the hypothalamus that is affected. So if the hyperactivity has occurred in the left hypothalamus, that will lead to a left-sided cluster headache, whereas if it's occurred in the right hypothalamus, that will lead to a right-sided cluster headache. So we have we have lots of things to explain. Firstly, I want to explain how does it actually cause the headache. We can then instantly justify why we get accompanying nausea and vomiting. We're also going to see why we don't get meningeal symptoms from how it generates the headache. And we then want to turn on to a discussion of where do the autonomic symptoms come from. And then right at the end, after we've discussed autonomic symptoms, we'll come back and discuss cutaneous allodynia. Okay, so how is it going to actually give rise to a headache? Well, it is believed that this hyperactivity in one side of the hypothalamus is then going to lead to activation of second-order neurons in the same side trigeminal-cervical complex. So we have been talking all the time about it occurring in the left hypothalamus, which is going to lead to a left-sided cluster headache. It's therefore going to activate the left trigeminal-cervical complex. And again, this is one of the points in this story that I'm afraid I can just draw an arrow. I can't really say exactly how this is going to work, but somehow the hyperactivity occurring in the hypothalamus is going to lead to activation of second-order neurons in the trigeminal-cervical complex on the same side. So it's going to come down here and activate the TCC, the trigeminal-cervical complex. So the trigeminal-cervical complex, remember, it contains cell bodies of second-order neurons that are going to carry nociceptive information up to the contralateral thalamus. So the left-sided trigeminal-cervical complex, it will have loads of second-order neurons that are going to cross over and go up to the right thalamus and then deliver and then of course third-order neurons will deliver the information from the right-sided thalamus to the right primary somatosensory cortex. So it's going to activate second-order neurons directly. It's going to bypass the primary nociceptive afferents, basically. And this is then going to be what gives rise to the headache on the left-hand side. Now, thinking back to the site that is usually affected, it's going to be in the frontal region, the periorbital region, and maybe even the temporal region. So these regions, frontal and periorbital regions, those are the jurisdiction of the ophthalmic division of the trigeminal nerve, whereas temporal region, the anterior temporal region usually, the bit that's very close to the eye, we know that that's the domain of both the maxillary nerve and then further back the mandibular nerve. So to get it in those sites, it must be somehow activating specifically the second-order neurons that are being synapsed onto by primary nociceptive afferents from these regions, hence how the pain would end up being perceived as being in those regions, because you're activating the second-order neurons that service the primary nociceptive afferents that service these regions. Okay, so that's believed to be how it causes this unilateral headache, by activating the unilateral trigeminal-cervical complex. And I think very importantly to put onto this picture is to put this word ipsilateral. So whichever side of the hypothalamus that the hyperactivity is occurring, and in this case we have drawn it on the left-hand side, it will then activate the same-sided trigeminocervical complex, in this case the left-sided trigeminocervical complex, which receives information from the left side of the head. It receives nociceptive information from primary nociceptive afferents from the left side of the head, and it's specifically going to activate second-order neurons that have primary nociceptive afferents from the ipsilateral, frontal, periorbital and temporal regions synapsing onto them, and that's how the pain is then going to be perceived as being in those regions on the ipsilateral side. And just on one side, if it activated the right trigeminocervical complex as well, then you would get a bilateral headache, but it doesn't generally. It usually just activates the ipsilateral side. So we have now explained why you're going to get a unilateral headache, why it's going to be in this region, and why, it's going to be very severe and feel as though it's stabbing because you're going to have massive activation of those second-order neurons. So in order to generate huge severity, you need to get these second-order neurons firing at a very fast rate. We have a huge hyperactive portion of the hypothalamus here. It's plausible that it can get those second-order neurons to fire at a very fast rate, hence how it's going to create severe stabbing pain as though someone is driving a knife into your eye. And it will presumably last the amount of time that the hyperactivity lasts. So presumably that means that the hyperactivity lasts usually between 15 minutes to 3 hours. Okay, so that's where the headache comes from. Now let's move on to some of these associated symptoms. So nausea and vomiting we understand. Whenever the cerebral cortexes, or cerebral hemispheres rather, are experiencing pain, they have pathways that are not well understood, linking them down to the vomiting centre in the brainstem reticular formation, and they can lead to activation of that vomiting centre, and depending on how much they activate that vomiting centre, you'll either just feel nauseous or you'll actually vomit. Why do you not get meningeal symptoms? Well, we can understand that as well. We are not actually activating meningeal nociceptors. Even though we might be activating the second-order neurons that meningeal nociceptors might feed into, because remember, often the primary nociceptive afferents from the meninges, they synapse onto the same second-order neurons as primary nociceptive afferents from peripheral sites, and indeed that was the main reason that we got the pain from the meninges referred to these regions in a migraine headache. Even though they activate the same second-order neurons, remember, we don't really understand the mechanism by which you get the meningeal symptoms, but i.e., even though the first-order neurons synapse onto the second-order neurons, somehow the brain still can tell the difference between peripheral nociceptor activation and meningeal nociception. And remember, I couldn't tell you the mechanism that underlies that, but it does seem that unless you have the actual activation of the primary nociceptive afferents from the meningeal nociceptors, that you don't actually get meningeal symptoms. And indeed, we haven't got those the activation of meningeal nociceptors, and therefore it makes sense for us not to have meningeal symptoms, even though we are activating potentially the same second-order neurons that would have been activated if the primary nociceptive afferents from the meninges had been activated. So what that tells us is that it must have some other pathway that we don't understand of knowing when the pain is actually coming from the meninges to produce the meningeal symptoms, but I can't answer what that is. Okay, so because we don't have activation of meningeal nociceptors, we don't get meningeal symptoms. We are activating directly the second-order neurons. No meningeal nociceptors will be activated. What we now want to turn to is the bit that's going to be complicated, the new bit, and this is going to involve us going over some more background anatomy. We need to try and justify the autonomic symptoms, which are going to be on the same side as well. So we're following this story for the left-hand side, so we're going to get now left-sided autonomic symptoms. We might get a left-sided ptosis, we might get left-sided meiosis, left-sided lacrimation, left-sided rhinorrhea, and a left-sided red eye, and we need to try and understand where those are actually going to come from. And I'm afraid, again, this is going to involve holes in the explanation, but to try and get as good an understanding as possible, we will look at the background anatomy that is understood. Okay, so where do the autonomic symptoms then come from? Well, this hyperactivity in the hypothalamus has been capable of directly activating these second-order neurons in the ipsilateral trigeminocervical complex. Isn't it now plausible that this hyperactivity in the hypothalamus here is also going to be capable of activating autonomic or inactivating autonomic innervation to certain areas on the same side as well, on the ipsilateral side? Indeed, we've discussed earlier when we were doing a description of cluster headaches that the autonomic symptoms really are all about activation of the parasympathetic nervous system innervation to the eye and the lacrimal gland and the nose, whilst you're also going to inhibit the sympathetic nervous system innervation to the eye as well, which is what results in the ptosis and the miosis. Now, the hypothalamus, I've said that this is an evolutionarily ancient portion of the brain and therefore controls very old portions of the nervous system, very fundamental, foundational parts of the nervous system. The autonomic nervous systems, the parasympathetic and sympathetic nervous systems, these are ancient parts of the nervous system, and actually they are controlled by the hypothalamus. And it's the same side of the hypothalamus that controls the ipsilateral autonomic nervous system, by which I mean the left hypothalamus controls the left-sided autonomic nervous system, and the right-sided hypothalamus controls the right-side autonomic nervous system. So, if we've got hyperactivity, this strange brain phenomenon occurring in the left-sided hypothalamus, it is very plausible that we can then get disturbances to the left autonomic nervous system. And indeed, we're going to get increased activation of the parasympathetic nervous system to the eye and the lacrimal gland and indeed the nasal cavity, and decreased sympathetic output to the eye. Now, again, it's not well understood the actual pathways that link the hypothalamus to the preganglionic parasympathetic and sympathetic fibres that go to the eye and the lacrimal gland and the nasal cavity. So all I can draw there is an arrow, but what I do want to now go over is the background anatomy of the preganglionic and the postganglionic neurons that are going to innervate these important tissues, so the eyes, the lacrimal gland, and the nasal cavity. Okay, so to spell out where we're going now, we're going back to look at some background anatomy. I want to discuss the sympathetic and parasympathetic innervation of the eye. Both of them are innervated by the sympathetic and parasympathetic nervous system. And then I want to discuss the parasympathetic innervation to the lacrimal gland and the parasympathetic innervation to the nasal cavity. I want to go over that anatomy. So we'll begin with the parasympathetic nervous system. And for this, it is necessary to discuss two brainstem nuclei that we haven't previously discussed, namely the Edinger-Westphal nucleus and the superior salivatory nucleus. And this will involve discussing two more cranial nerves that we haven't previously discussed, namely the oculomotor nerve and the facial nerve. So we will begin with cranial nerve number 3, the oculomotor nerve, and the Edinger-Westphal nucleus. And this is going to deliver parasympathetic innervation to the eye, and it's going to be responsible for causing the miosis. It's one of the things that's going to be responsible for producing the constricted pupil. We'll also see that it's responsible for making the lens more globular, causing the contraction of the ciliaris muscle. So, background information now. We want to study parasympathetic innervation to these tissues. We're going to start with parasympathetic innervation to the eye, and then once we've done all of that, background anatomy of the autonomic nervous system innervation to these tissues will then complete up our story of the pathophysiology underlying cluster headaches. Okay, right. So, cranial nerve number 3. I'm going to write this down here. We're going to study cranial nerve number 3, CN3, also called the oculomotor nerve. And particularly, we want to discuss the parasympathetic fibres that run in cranial nerve number 3. Okay, so firstly, I want to have a look at the nucleus of the oculomotor nerve and the Edinger-Westphal nucleus, and we will see this in a cross-section of the medulla. Now, we have got rid of our previous cross-section, sorry, not the medulla, in a cross-section of the midbrain, a rostral cross-section of the midbrain. Now, we have got rid of our previous picture of the cross-section of the midbrain, so I'm now going to draw that again. So just bear with me, it'll be good revision. We can revise the previous structures that we've seen in the rostral section of the midbrain. So remember, rostral midbrain means I'm taking it right at the top here towards the nose, and this is the identical picture that we drew earlier, but now I want to show you the oculomotor nucleus and the Edinger-Westphal nucleus. So, again, we will put the back of the midbrain at the top, and we'll put the front of the midbrain at the bottom. So those are the superior colliculi, and this is going to end up being a big picture. Here we have the, which way around am I doing it? That will be the right cerebral peduncle, because I'm imagining we're viewing from above. Then we have the left cerebral peduncle here, and then just connect that up like so. So we're looking from above, just to orient you. This is the right side, this is the left side, this is the front, and this is the back. Okay, so let's now put some of the fancy structures that we saw earlier on. So test yourself. Shout at your computer screen what I am drawing on at the moment. So what is this? This is, of course, the periaqueductal grey with the cerebral aqueduct running through it. Now let's put on the substantia nigra, again in a bright colour because it's the only thing that will show up. So here is the left substantia nigra, and here is the right substantia nigra. And of course in front of the substantia nigras we have the cerebral peduncles, where all of those axons are running down from the cerebral cortex. So we have the corticospinal tracts running through there, and the cortico-pontocerebellar pathways running down there as well. Okay, next, let's put on the medial lemnisci. So here's the left medial lemniscus just behind the substantia nigra, and here's the right medial lemniscus. Then more things, the trigeminophalamic tracts, of course, are medial to the medial lemnisci, so there they are. And then the spinothalamic tracts are other things that we've seen, so I'll put those on. Those are lateral to the medial lemnisci in these positions here. So those are the spinothalamic tracts. And then we need to put on the red nuclei, and I'll get a very red pen for putting the red nuclei on. So here are the red nuclei. We would have the reticular formation. I'm not actually going to put the reticular formation on because it will get in the way of where I want to show other things. But of course the reticular formation will be in all of this sort of space here. And of course the other thing that we saw earlier on is the mesencephalic nucleus of the trigeminal nerve, which is back there at the back of the reticular formation at the midbrain level here. Okay, so excellent. There is my rostral cross-section of the midbrain. We've nicely revised the structures that we've previously seen. Now what I want to show you is the oculomotor nucleus, the nucleus of cranial nerve number 3. So I will put this in a new colour. We'll have orange. So it's in this sort of position here, and this is why I didn't want to put in the reticular formation, because I thought it would get in the way of me showing this. The reticular formation is here, but it's in the front of the oculomotor nuclei. Here, the oculomotor nuclei are just in front of the periaqueductal grey here. So I'll label these up because these are new structures. So in orange here, these are oculomotor nuclei, cranial nerve 3's nuclei in orange there. Right, and now let's get another new colour. I'll have some new yellow here. And I want to now put on the Edinger-Westphal nuclei, which are just behind the oculomotor nuclei. So in that mustard colour there, those are the Edinger-Westphal nuclei. Also for short, often abbreviated down to the EW nuclei. So you can call them the EW nuclei if you wish. Right, so these nuclei are going to have the cell bodies of neurons that are going to go into the oculomotor nerves, which are going to come off the front of the midbrain in between the two cerebral peduncles here. Now, what do the oculomotor nerves do, in case you haven't heard of these nerves before? These are the nerves that move the eyes, hence why they're called oculomotor nerves. They contain the neurons that are going to move the muscles of the eyes. They contain the motor neurons that go to the muscles of the eyes. They also contain the preganglionic parasympathetic fibers that are going to go to the eyes. Remember, whenever we're discussing the autonomic nervous system, both sympathetic and parasympathetic divisions, when you want to innervate a tissue with autonomic supply, you have a preganglionic fiber coming out of the central nervous system, and then it doesn't synapse directly onto the structure. It then synapses onto a postganglionic neuron, which then delivers the information to the tissue. Remember, with alpha motor neurons, it's just one neuron comes out of the central nervous system and innervates the tissue directly. The autonomic nervous system is slightly more faffy around. It has two neurons, one relays it onto the other, preganglionic and postganglionic. We'll see how this works in the examples in just a moment. Okay, so the oculomotor nerve nuclei in orange here, these are going to contain the cell bodies of alpha motor neurons that are going to innervate the muscles of the eye. And of course, the left oculomotor nucleus here will contain alpha motor neurons that will go to the left eye, and the right oculomotor nucleus will contain alpha motor neuron cell bodies that will go to the right eye. So let me now put on the oculomotor nerves. So here they come, forward like so, and you can see these on the cross section of the midbrain. So they'll be in these sort of positions, and they go through all of these structures, and you can see them emerging from the front here. So these are our cranial nerve frieze. So we have the left oculomotor nerve and the right oculomotor nerve coming out in between the two cerebral peduncles here. And these will be where the axons of the alpha motor neurons are running. So alpha motor neurons that are going to innervate, let's say we're looking at the right side here, right-sided orbital muscles will have their cell bodies here in this right oculomotor nucleus here, and they'll send their axons out in the right oculomotor nerve, and this will go forward to actually innervate the muscles. The Edinger-Westphal nucleus, this is a parasympathetic nervous system nucleus, which is why we're studying this at all. This will contain cell bodies of preganglionic parasympathetic fibers. So it's only parasympathetic, not sympathetic. So this is a parasympathetic brainstem nucleus. So these are going to be preganglionic because they're going to synapse onto the postganglionic fibers in a ganglion. So these are preganglionic parasympathetic nervous system fibers. Okay, right, and these are going to be fibers that are going to supply the ciliaris muscle and also the sphincter pupillae muscle. So in there will be cell bodies of preganglionic fibers. These are also going to be multipolar neurons, so their cell bodies will be in there, and then their axon will go out into the ipsilateral cranial nerve here. So if we're talking about the right side, it will send its axon into the right oculomotor nerve, which will then go into the right orbital cavity. Okay, right, so that's the Edinger-Westphal nucleus and the oculomotor nerves emerging. The oculomotor nerves, they're then going to have to run forward. Remember where the midbrain is. It's just above the pons, and therefore it's just at that forefront of the superior portion of the posterior cranial fossa. So let me just draw another little picture that we've seen again many times before, which is going to be a picture of the cranial cavity, just to show you where the oculomotor nerves will run. So here we go, there's the oval, familiar oval. Here is the anterior cranial fossa, here are the clinoid processes, the sella turcica here, and of course the petrous portions of the temporal bone, and then there is foramen magnum. Right, so remember the midbrain is in this sort of position above the tentorium cerebelli level. Below the tentorium cerebelli you then have the pons and the medulla. Okay, so off the front here are going to come the oculomotor nerves, and they're going to run like this on either side of the hypothalamus. So here they come in pink here. I'll just draw them in a little bit thicker so that they actually show up. So here is the right one there, and here is the left one. And you can probably guess what's going to happen here. They are coming off the central nervous system, so they're going to now be outside of the central nervous system. They are counted as peripheral nerves. So they're going to penetrate through the pia mater that covers the brain, and they'll then end up in the subarachnoid space. And what are they running on top here? They're running on top of the cavernous sinus. And of course, the top walls of the cavernous sinus will be meningeal dura mater covered by arachnoid mater. And they actually, again, like the ophthalmic nerve and the maxillary nerve that we discussed previously, they're going to run in that seeding of the cavernous sinus in the meningeal dura mater and the arachnoid mater. So they run over the cavernous sinus, and they run more medially than the ophthalmic nerve and the maxillary nerve. So the ophthalmic nerve will be here, and the maxillary nerve will be here, more lateral to the oculomotor nerves. So the oculomotor nerves are then going to go through the superior orbital fissure, and of course, to get through the superior orbital fissure, they'll have to penetrate through the outer meninges, right through the periosteal dura mater to go through the bony hole, and they'll then end up in the orbital cavity. So they go through the superior orbital fissure. Right, so the question is, what happens then once we're in the orbital cavity? Well, the oculomotor nerve is going to split into two parts. So let me draw this here. So again, I'm going to take the right-hand side. We've always been discussing the right-hand side, even though actually I've just realised that I've been a little bit inconsistent there, because when we were discussing the hypothalamus, we were discussing the left-hand side, and now we've moved on to the right-hand side for cranial nerves. But for these pictures, I prefer drawing the right-hand side for some reason. Sorry about that. We'll have to go back to the left-hand side when we want to actually reconcile the pathophysiology with this background anatomy, but never mind. Okay, so right-hand side now. So here is the actual eyeball here. And this is the same picture that we drew long ago, where we showed the anatomy of the ophthalmic nerve and the anatomy of the ophthalmic artery, but we won't put that all back on now. So we want to see the oculomotor nerve. So here comes the oculomotor nerve, and I think I'll get a thicker pen rather than having to colour in. So here comes the oculomotor nerve. It's arrived in through the superior orbital fissure. We don't have to worry about any complicated anatomy with the meninges anymore, because it's penetrated through the outer meninges and it's now outside of the cranial cavity. It then splits into two branches. So firstly, it's going to split into two branches like so. A superior branch that's going to go up, and an inferior branch that is going to go down. So it's difficult to show this on this picture where we're looking from above, but this one is representing the superior branch, and this one is representing the inferior branch of the ophthalmic nerve. And remember, oculomotor nerve. And remember, the oculomotor nerve carries two types of fibre, the axons of the alpha motor neurons and the axons of the preganglionic parasympathetic nervous system fibres. Alpha motor neurons are going to go both ways, but parasympathetic fibres are going to go into the inferior branch. So let's discuss the superior branch first, just to get that out of the way, and then we'll go on to the inferior branch, which is the one that obviously we care about more. So the superior branch will split into two branches then, and these two branches will supply the two superior muscles of the eye, of the orbital cavity, which are the superior rectus muscle, and I haven't gone back to a thinner pen, the superior rectus muscle, and also the levator palpebrae superioris muscles. So let me write out their names in full. So SR stands for the superior rectus muscle. So that branch will go to the superior rectus muscle, and that will contain alpha motor neurons from the oculomotor nerve nucleus. And then LPS stands for the levator palpebrae superioris. And this means that it's going to elevate the eyelid, basically. Levator palpebrae superioris. So let me just sort of demonstrate the anatomy of these muscles to you. The superior rectus muscle, it's in this sort of shape. So it's a muscle that's in this shape that I'm tracing out here. It attaches to the back of the orbital cavity here. It attaches to the bone at the back, and then it extends forward to attach to the top of the eyeball. And when it contracts, it's going to pull the top of the eyeball backwards, and therefore it's going to elevate the eyeball. It's going to make you look upwards. It's going to move the eyeball upwards so that the pupil is now looking more superiorly. So the superior rectus, that elevates the eyeball. Levator palpebrae superioris doesn't actually move the eyeball, it moves the eyelid. Again, it's a muscle that goes forward, it's attached to the back and it goes forward, but it attaches to the eyelid tissue, and when it contracts, it pulls the eyelid upwards. Now, the alpha motor neurons that are coming to the levator palpebrae superioris from the oculomotor nerve nucleus, they are going to innervate the skeletal component of the levator palpebrae superioris. So the levator palpebrae superioris is a complicated muscle. It has two components. Some of it is skeletal muscle, and some of it is smooth muscle. And it's the skeletal muscle that, of course, alpha motor neurons are going to innervate and which you have conscious control over. So when you raise your eyelids open, this is the portion of the levator palpebrae superioris that you are using. When you're consciously doing it, this is how you're doing it with the skeletal muscle of levator palpebrae superioris. When you're not thinking about consciously keeping your eyelids open or moving your eyelids, but they remain open anyway, that is because the smooth muscle component is being contracted by sympathetic fibres, and we'll come on to those sympathetic fibres and how they're going to get here later on. With regards to discussing the oculomotor nerve, all it's going to innervate for its alpha motor neurons is the skeletal component. We'll come back to sympathetic component much later. We want to do parasympathetic nervous system first. Okay, right, so that's levator palpebrae superioris and superior rectus. Now onto the inferior branch, which carries the preganglionic parasympathetic nervous system's axons. So the inferior branch is going to split into 3 branches this time. So it's going to split into one branch that will go to the medial rectus, one branch that will go, and these are underneath, you have to imagine they're underneath the eyeball, these. One branch that will go to the inferior rectus, and then one branch that will go to the inferior oblique. So imagine I have drawn them underneath the eyeball rather than they look as though they're on top of the eyeball. Okay, so this one here, this one's going to the medial rectus, and this muscle is in the medial portion of the orbital cavity. Again, it extends backwards and attaches on back here at the back of the orbital cavity, and it attaches at the eye over here, and when it contracts, it will pull the eye round in this direction, and therefore will adduct the eye. It will make you look towards the centre rather than looking outwards. Okay, so that's the medial rectus, and this will be supplied by alpha motor neurons. It's a skeletal muscle, a voluntary muscle. So alpha motor neurons will go down that branch. Other 2 branches, one is going to go to the inferior rectus muscle. We'll start with that one, and it's getting a little bit crowded, this picture now. This one is meant to be the one going to inferior rectus. Inferior rectus is effectively the opposite of superior rectus. Imagine the same sort of shaped muscle, but underneath the eyeball. Again, attached to the back of the orbital cavity, the bone, but attached to the bottom of the eyeball. And when it contracts, it'll pull the eyeball down so that you look down. So it will depress the eyeball rather than elevate the eyeball. So superior rectus elevates the eyeball, inferior rectus depresses the eyeball. And again, it's going to be innervated by alpha motor neurons. So all alpha motor neurons are going to go down there. So all of these branches that I've seen so far, they're just the alpha motor neurons coming from the oculomotor nuclei. The parasympathetic fibres are actually going to come down this branch, the inferior oblique branch, as along with, again, more alpha motor neurons from the oculomotor nuclei. So this is the inferior oblique, and this one's got a different name, so I suppose I should probably write its name out in full. So IO stands for inferior oblique. I think that's how you spell it. Okay, so inferior oblique is another skeletal muscle. It's in a different position, however. It actually attaches to the front of the orbital cavity, and this is the bottom of the orbital cavity, so it's underneath the eyeball. It's underneath, in fact, inferior rectus. So underneath inferior rectus, the inferior oblique runs like this, in this sort of shape that I'm tracing out now. It attaches to the bottom medial corner, bottom medial anterior corner of the orbital cavity. It then extends backwards to attach to the eyeball back here, and when it contracts, it does 2 things. Firstly, you can imagine that will actually elevate the eyeball. If you're pulling the bottom of the eyeball back here forwards in that direction, that's actually going to make you look upwards. Think about this carefully. The inferior oblique is a complicated muscle, so it's going to make the eyeball look upwards, so it will elevate the eyeball. In addition, it's actually going to spin the eyeball around, so it's going to, imagine pulling this bit here backwards up here, that will end up pulling the eyeball this way round in that sort of an arrow direction. And therefore the eyeball will end up looking lateralward, abduction of the eye. So inferior oblique actually makes the eyeball look upwards and outwards. Complicated muscle. So in this branch from the inferior branch of cranial nerve 3, and I would just like to actually label this up as being cranial nerve 3 here, from this inferior branch of cranial nerve 3, the branch going to the inferior oblique will contain alpha motor neurons that innervate the inferior oblique muscle and make you look upwards and outwards in this right-hand eye here. However, this branch to inferior oblique is also going to contain the axons from the Edinger-Westphal nucleus, the preganglionic parasympathetic fibers. And these are actually going to come off this branch and go into the ciliary ganglion. And this is where the ciliary ganglion actually gets fibers that are going to synapse in it. So remember we discussed the ciliary ganglion long ago, so I'll put it back in on this picture there as that mint green blob. So this is the ciliary ganglion. So we discussed this long ago when we were discussing the ophthalmic nerve. Remember, branches came off the nasociliary nerve, which were containing sensory afferents, and they ran straight through the ciliary ganglion. And then the ciliary ganglion, remember, gave off the short ciliary nerves, which I'll now try and put in here. So those little things that I've put coming off the ciliary ganglion, those are the short ciliary nerves. So it was really totally pointless, the ophthalmic nerve, putting those branches into the ciliary ganglion because they didn't actually synapse in there at all. They just ran straight through it. However, here we are now going to see fibres that actually do synapse in the ciliary ganglion. So let me get another colour. So from the inferior oblique branch, here in vivid purple, and I want to get a thicker pen so it actually shows up. Here in vivid purple, that branch coming from the inferior obliques branch is where the preganglionic parasympathetic fibres are going to leave the inferior oblique branch and go into the ciliary ganglion. And in the ciliary ganglion, what's going to occur? There's going to be a synapse. Here is the preganglionic parasympathetic fibres axon terminal, and it will then synapse onto the postganglionic fibre. And here is my picture of the postganglionic one. So here in, again, vivid purple, this is the postganglionic parasympathetic fibre that will have its cell body in the ciliary ganglion. And this is why the ciliary ganglion is indeed a ganglion, because it contains these cell bodies of the postganglionic parasympathetic fibres. And they will then send their axons in through the short ciliary nerves here, which will penetrate through the sclera into that gap between the choroid and the sclera, and they'll then run in that gap forward, and they will innervate the ciliaris muscle at the front and also the sphincter pupillae muscle of the iris. And the one that we really care about is the sphincter pupillae muscle of the iris. So let me just draw a little picture of the iris. So here is the iris here. Here's the pupil at the centre, so that's the pupil, and then the muscle surrounding it is the iris. Okay, now the iris has two major muscles which are both under the control of the both under the control of the autonomic nervous system. One is under the control of the parasympathetic nervous system, and the other is under the control of the sympathetic nervous system. So the one that is under the control of the parasympathetic nervous system consists of smooth muscle cells that are oriented in circles like this. So the smooth muscle cells are connected to one another to form rings in the iris muscle. And this is called the sphincter pupillae muscle. Because if you activate those smooth muscle cells to contract, they're all going to contract, they will all become shorter, and therefore the circumference of the rings of the smooth muscle cells will become smaller, and therefore the diameter of the rings will decrease, and therefore they will constrict the pupil. So there are loads of smooth muscle cells ordered together to form rings around the iris, or around the pupil like so, within the iris. These are called the sphincter pupillae muscles, and if you activate their contraction, it will cause the iris to contract and close the pupil, i.e. it will cause miosis. Whereas the other muscle, which is controlled by the sympathetic nervous system, it has smooth muscle cells that are oriented in this sort of direction, radially outwards like so, and that is known as the dilator pupillae muscle, because if you activate the contraction of those smooth muscle cells, of course, they're going to pull the pupil open. They're going to cause mydriasis, the exact opposite of a wide pupil. So the sympathetic nervous system activates dilator pupillae, the parasympathetic nervous system activates sphincter pupillae, and is therefore going to cause pupillary constriction. The other thing that the parasympathetic nervous system is going to innervate within the actual eyeball is the ciliaris muscle, and it causes constriction of that ciliaris muscle, and therefore it causes the lens to become more globular, i.e. ideal for looking at things that are very close to you. However, that's not one of the important symptoms that you will notice when you have a cluster headache, so it wasn't one of the symptoms we were aiming to explain, so we won't go into that. Okay, so the postganglionic parasympathetic fibres, they'll go in through the short ciliary nerves and they'll get into that space between the sclera and the choroid, and they will innervate the sphincter pupillae muscle. And if you activate the preganglionic ones, then they'll activate the postganglionic ones, and therefore they will lead to miosis. So one of the things that the hypothalamus is somehow going to have to do in a cluster headache to give rise to some of these autonomic symptoms is to activate the ipsilateral Edinger-Westphal nucleus. So if we had this activity occurring in the left hypothalamus, it's going to need to activate the left Edinger-Westphal nucleus, and that will lead to miosis of the left pupil, where we are also feeling the horrible, horrible pain. Okay, so just to summarise what we have learned about the Edinger-Westphal nucleus and cranial nerve number 3. So we saw where the oculomotor nuclei were and where the Edinger-Westphal nuclei were. We have a left oculomotor nucleus and a right oculomotor nucleus, and we have a left Edinger-Westphal nucleus and a right Edinger-Westphal nucleus. The alpha motor neurons that innervate the muscles of the orbital cavity, most of the muscles of the orbital cavity, there are some others that aren't innervated by the oculomotor nerves, such as the superior oblique and lateral rectus muscles, but a lot of them are innervated by the oculomotor nerve, and the alpha motor neurons that innervate those are located in the oculomotor nuclei, and the left oculomotor nucleus serves the left orbital cavity and the right oculomotor nucleus serves the right orbital cavity. Preganglionic parasympathetic fibres that are going to serve the actual eyeball and which are going to cause the activation of the ciliaris muscle and the sphincter pupillae muscle, they have their cell bodies in this Edinger-Westphal nucleus on either side that is a parasympathetic brainstem nucleus. Okay, you have a left one and a right one, and they send their axons into the same-sided oculomotor nucleus, and therefore they serve, oculomotor nerve, and therefore they serve the same-sided orbital cavity. We've discussed how the oculomotor nerves are going to cross the middle cranial fossa in the wall, the top of the cavernous sinus, and then they're going to get into the orbital cavity by going through the superior orbital fissure. In the orbital cavity, they're going to split into a superior and an inferior branch. The superior branch gives two branches, the superior rectus branch and the levator palpebrae superioris branches, which contain the fibres of alpha motor neurons from the oculomotor nuclei. The inferior branch has three branches one goes to the medial rectus, one goes to the inferior rectus, and one goes to the inferior oblique. The preganglionic parasympathetic fibres, they go down the inferior obliques branch, and then they come off to go into the ciliary ganglion, this purple branch here, where they synapse onto the postganglionic parasympathetic fibres that are in the ciliary ganglion, and those postganglionic parasympathetic fibres will go in the short ciliary nerves to get into the eyeball and then innervate the ciliaris muscle and the sphincter pupillae muscle. Okay, so that's the background anatomy of the Edinger-Westphal nuclei. We'll have a break here, and in the next video we will move on to discuss the superior salivatory nuclei, another very important example of a parasympathetic brainstem nucleus that is going to supply the lacrimal glands with parasympathetic innervation, the nasal cavity with parasympathetic innervation, and also the conjunctiva with parasympathetic innervation, which is important for causing the red eye.